Thank you for your nice introduction and good morning, everyone. My name is Jiwon and I'm a pathologist in Seoul National University Hospital. I've been practicing cardiovascular pathology since the year 2020, and it is a great honor to have a talk here in APCIS this year. So my topic today is about the embryology and development of ventricular outflow tracts. And regarding the embryology, we all learned this during the medical school and we took exams on it. So it's a kind of classic topic, but it's very crucial to understand the embryology, to understand the pathophysiology of the cardiovascular disease. So to start with, I will begin my talk with the classical view on the heart embryology. And since after I've been preparing for this talk and studying about the embryology again, I found the very interesting perspective, more refined perspective regarding the cardiac development suggested by the Dr. Robert Anderson. And I would like to share his thoughts and his evidences using the light microscopy and electron microscopy figures, the wonderful figures. So let's start with the classical view. So the development of the heart starts after the gastrulation. There is a lesion in the embryonic disc called lateral plate mesoderm, like this, and this eventually formed the pericardial cavity. In a subset of this lateral plate, there exists a region called heart-forming region, or HFR, which is lined blue here. And as the embryo begin to undergo lateral folding, this blue HFR eventually makes a tube-like structure. So we call this primitive heart tube. And this cylindrical tube eventually goes dilatations, and this is a very interesting figure. This blue dye is it labels the myosin light chain. So theoretically, this labels the HFR region. So if we are looking at the embryonic disc right here, we can see this crescent-shaped heart-forming region. And the heart-forming region is regulated by a transcription factor called NKX 2.5. So after the heart tube is made, it goes into the dilatation phase. So as the previous speaker nicely introduced, the heart tube is composed of five components, starting with the truncus arteriosus and bulbus cordis, and primitive ventricles, and primitive atrium, and sinus venosus. So we all learned this as a medical student, and we all learned that the counterparts of adult structure, and we all took exams on it. So one of the refined view by the Dr. Anderson is based on the problematic description, like does the truncus arteriosus and bulbus cordis, can these two structures divided correctly, or it's kind of some kind of continuous spectrum? So that kind of perspective we will discuss later on this talk. And the previous, these previous heart tubes are going on to the next step. The next step is the cardiac loop formation. So as we all learned from the embryology class, the cranial portion of the cardiac tube bends ventrally and caudally to the right to form this structure. And while caudal portion shifts dorsocranially and to the left. So this is the ball-like structure made by looping of the cardiac tube. And the most important topic will be aortopulmonary septum for this session, because it is the crucial for development of the great arteries. And theoretically, according to the classic perspective, this AP septum is made from the neural crest cells migrating from the hindbrain region. So these neural crest cells invade into the truncal and bulbar ridges, and it grows spirally within this tube, and it eventually divides the truncus arteriosus and bulbus cordis into aorta and pulmonary trunk. So this idea of a spiraling septum, as a medical student, when I was learning this topic, it was really hard to understand, and I couldn't feel what is the nature of this spiral. So it was kind of problematic, and Dr. Anderson's view excellently explains how this AP septum, in fact, exists in the histological sections. And the clinical correlations we have to see here is that some of the major conotruncal anomalies can be inferred from the abnormal development of AP septum. For example, the persistent truncus arteriosus would result from a partial development or no development of AP septum. And complete TGA would be caused by non-spiral AP septum. So if the spiral growth is restricted within this trunk, the division of the two great arteries cannot be complete. So, and the TOF would result from the skewed development of the AP septum, not causing some kind of deviation to the right or left. So let's move on to the Dr. Anderson's wonderful pictures and perspective. So this is the picture of the primitive heart tube. Originally, this primitive heart tube is a kind of inverted Y shape, and it is symmetrical at the very early stage of the development. This is the ventricle, and we can see the outflow tract here. And venous pulse consists of the two inverted arms of the alphabet Y. And we can see here by labeling the myosin light chain by blue dye in this embryo. And we are seeing the embryo in the lateral view, and we can see that this area is supposed to be the outflow region, but it's a little bit paler than these ventricle or atrial structures. When we label this embryo using FGF10, we can see that this initially pale area of the outflow tract is stained. So using this evidence, Dr. Anderson suggests the concept of secondary heart field, and the secondary heart field would be responsible for the development of outflow tract and a part of RV structure. This is another supporting evidence with a higher magnification, and we can see that FGF10 labeling of the secondary heart field, we can see it is labeling the pharyngeal arches, the outflow tract, and developing RV. And the left ventricle, it's supposed to be here, and it is not stained at all. It means that cells from the secondary heart field do not extend beyond the interventricular group. And it was a very interesting concept for me. Another evidence is that if we mark red color into the cranial pole of the initial heart tube, this eventually, after the development, this marker goes into the left ventricle and the interventricular group, which means that the more distal part of the RV and outflow tract have been recruited from the secondary heart field, not from the initial primitive heart tube. So regarding the looping of the heart tube, this is another excellent example of electron microscopy. And we can see the tube bending to the right, and apical part of RV will balloon from this inlet, and RV will balloon from this area. And regarding the outflow tract, it will be continuous with the aortic sac regulated by these genes. As the days develop, this is the more developed outflow tract here, and we can see this acute bending. We call this dog leg bend, and based on this angle, we can divide the outflow tract as proximal and distal. So from this phase, initially these two distal and proximal outlets are encased by myocardial tissue initially, but it progressively goes into arterial phenotype. And the distal part will be developed into intrapericardial portions of aorta and pulmonary trunk, and the proximal part will be valvular leaflets, and more proximal parts, it will be the subpulmonary infundibulum. And let's go into the problematic structure called AP septum. So proximal and distal parts are angulated by this dog leg bend here, and this is that angle. And one interesting perspective by Dr. Anderson was that this lumen is lined by a layer of endocardial jelly. And it eventually concentrates into facing cushions lining the lateral wall of this outflow tract. And these endocardial cushions are very responsible structures for formation of AP septum. So this endocardial jelly enables the spiraling growth of the septum. And as the days developed, we can see the changes here. And this star highlights the development of endocardial cushion from the endocardial jellies on the walls. And this cushion divides this part of the outflow tract into the aorta and pulmonary trunk. And this is another example of the distal cushion. And for the population of the densely stained cells right here, is some of the endocardial cushions, they include some cells migrated from the neural crest. And this is another frontal view, highlights the divided structures of distal outflow tract into aorta and pulmonary trunk. So the distal cushions have divided these two structures. And this structure, the distal part, eventually, right now, has the arterial phenotype, not the myocardial phenotype. And this cross-sectional view also shows the divided aorta and pulmonary arteries by the endocardial cushion highlighted here. And this structure, the distal part, is now arterialized, but the proximal outflow tract still has its cushion on its lumen, and it is composed of muscular walls, the myocardialized walls at this stage. But as the time goes by, the proximal outflow tract also undergoes arterialization by the contribution of cushions and neuroclast-derived cells. So finally, almost finally, the division of the outflow tract is almost complete, and we can see these endocardial jellies and cushions are starting to make the valves of these great arteries. And two great vessels are divided completely right here. And this is a very, this is the very adult-like, almost developed heart. And we can see that proximal outflow cushions are, finally, this is the only endocardial cushions remaining in the proximal outflow tract, and it's located between the pulmonary valve and tricuspid valve as a supraventricular crest. So this was the outline provided by refined view. So conventional idea was the five-part composition of the primitive heart tube, subsequently dividing and dilatating and folding and looping. But the refined view of the ventricular outflow tract includes the concept of secondary heart field in the development of outflow tract and a part of RV. And it divided the ventricular outflow tract into distal and proximal part using the dog-leg band as a boundary. And it describes the mysterious transition from the myocardial walls of the outflow tracts into arterialized phenotype. And we still don't know what causes this mysterious arterialization. And apart from the theoretical concept of AP septum, it is suggested that endocardial jelly or cushions and some of the neural crest developed cells actually makes the cushion at the lateral wall of the developing outflow tract, and it is the structure which enables the spiral growth of the AP septum. So this is the end of my talk, and thank you for your attention.